food has been largely minimized. But more recently, let's say in the last 15 years, this approach has been complemented and it was proposed to pay increasing attention to the concept of food processing. A milestone in this sense was the NOVA classification developed by a group of Brazilian research which divided food into 4 groups basing on the degree of the processing. So in the first group we find unprocessed or minimally processed food, which are food that did not undergo some process such as fruit and vegetables, but also eggs or meat. In the second group we find processed culinary ingredients generally used to cook, such as salt or sugar or oil. In the third group we find processed food, which are food from first group plus processed culinary ingredients. Example are cheese or bread or buttered vegetables. Finally, in the last group, the first group, we find ultra-processed food, which are mostly industrial formulation made with intense chemical or physical process, often with added of colors, flowers or other cosmetics additives. Example of ultra-processed food are processed meat, soft drinks, but also food that are considered nutritionally healthy, such as some kind of breakfast cereals and fruit yogurt. For the purpose of this thesis, we used the data from the Moli-san study, a large prospective cohort study which enrolled from 2005 to 2010 around 24,000 subjects aged more than 35 years from the general population of the Molise region in southern of Italy. First of all, we have to make an important difference between chronological age and biological age. Because chronological age is the number on your passport, but it's not able to reflect the heterogeneity between each person during the aging. For this reason, in the last decade has gained increasing attention the concept of biological age, which is the hypothetical underlying age of an organism and may predict mortality and morbidity better than chronological age. The discrepancy between biological age and chronological age is called biological aging and can assume positive or negative values. Just to explain the difference between chronological and biological age, on the left on the screen you can see the chronological age of a subject, which is related with the birth date of a subject. On the contrary, on the right of the screen you can see the biological age of a subject, which reflect how the body aged. There are different biomedical parameters used to compute biological age. Among these we find the DNA methylation, neuroimaging, spirometry or blood biomarkers. In our cohort, to compute biological age and consequently biological aging, we used the blood-based approach because it's relatively easy to obtain and it can be computed, replicated in other cohort. In particular, we use a deep neural network, an algorithm which works like human brain. In particular, we have input layers, the 36 blood biomarkers, a variable number of intermediate layers where the information is processed, an output layer which corresponds to the label of the algorithm, in our case the chronological age. As a result of our algorithm, we obtain the biological age, and the discrepancy between biological age and chronological age was computed for each participant. In particular, to compute biological age in our cohort, we started from the whole sample of the Moli-san study, and after passing a quality control, our sample was around 23,000 subjects. Here we can see the 36 blood biomarkers used to compute biological age in the Moli-san study cohort. In particular, these biomarkers reflect the main biological pathway associated with the chronic disease onset and progression. Among these we find markers of inflammation, markers of renal or cardiovascular function, markers of glucose metabolism and lipid metabolism, and so on. As we said, biological aging can assume a positive or negative value. In particular, positive value of biological aging means an acceleration in aging process. On the contrary, negative value of the biological aging leads to deceleration in aging process. As we can see in this slide, negative value of biological aging are associated with the lower risk of all-cause mortality. Now, going on to the results of AIM-1. And in relation to the first aim of this thesis, we examine the adherence to a traditional Mediterranean diet as a gold standard of diet quality. In particular, our results show that the increasing adherence to this dietary pattern was associated with the lower value of biological aging, suggesting that people who following a Mediterranean diet are biologically younger. As an additional measure of diet quality, we examined the impact of food rich in polyphenols on biological aging. In particular, as we can see, the higher the consumption of food rich in polyphenols, the slower biological aging. Now, going on to the results of AIM-2, another key findings in this analysis is the positive association between ultra-processed food consumption and blood-based measure of biological aging. Basically, people who consume many ultra-processed food in their diet are biologically older. Importantly, this association was found to be independent by overall diet quality. Indeed, when we added the Mediterranean diet score in the analysis, the association between ultra-processed food and biological aging decreased around 9%, suggesting that other characteristics and the food nutritional characteristics, the poor nutritional characteristics of this food, could play a role in the association between ultra-processed food consumption and biological aging. In conclusion, the findings of this thesis provided an additional evidence on the positive effect, beneficial effect of healthful dietary pattern, such as Mediterranean diet, on biological aging, highlighting the contribution of food rich in polyphenols. Moreover, this thesis provided the very first evidence on the adverse effect of ultra-processed food consumption on blood-based measure of biological aging beyond their poor nutritional characteristics, shedding a light on the other potential mechanisms to take in account when we examine the impact of the diet on aging process. Thank you for your kind attention. I would to leave you with this sculpture, Donna che trattiene il tempo, by Donato Barcaglia, currently in Museo Volterra in Trieste. And this sculpture shows a woman, a symbol of youth, who tries to hold back time, sculpted like a winged old man who is running away. Thank you again for your attention, and I leave the word to the prorector.
Thank you for your presentation. The opposition will be opened now by Professor Brouwers, Professor of Internal Medicine and especially endocrinology and metabolic disease at this university, and Professor Brouwers was also a member of the assessment committee.
Thank you. So, dear candidate, before I start, first I would like to congratulate you and your team for this very nice thesis, also very nice cover. You know, in the Netherlands we don't have these nice sculptures, so it's really nice to see a different type of cover in this thesis. Again, I enjoyed reading the thesis. I found it very interesting to read it. But while I was reading, I got more and more, well, I don't think it's really confused, but I really wondered about the term biological aging. What is it? You already, in your introduction, told something about it, but if you could give a definition of biological aging, what would it be?
So, I listen to the opponent. Thank you for your congratulations, first of all, and thank you for your question, because it's one of the, of course, of the topics of this thesis. So, as I said, in the presentation. Biological aging is, first of all, the discrepancy between biological age and chronological age. So, as we say, the biological age, biological age is the hypothetical age of an organism, is how the body age. Of course, it could be different by chronological age. And the discrepancy, the difference, numerical difference, is an index of biological aging, which is very different from the chronological aging, and it could be very important. So, in order of health publicity, so for example, public health. So, because chronological aging is equal for everyone. We get older year by year, every year, 1 year for every year. Biological aging reflects how the body, the organism, the organs or the tissue aging. Yeah. So this is the difference. And sorry.
Sorry to interrupt you. So in your study you mostly focused on dietary aspects in relation to biological aging, but what do you think, are there also other determinants of biological aging?
Yeah.
What could that be?
Yeah. You have to imagine that the around 48% of the variance in biological aging is explained by a difference in lifestyle and socioeconomic status. Mm-hmm. Beyond the diet, in our group, we find that BMI, for example, smoking status, physical activity, and socioeconomic status can influence the aging, the variance of biological aging. Moreover, there are explanation in the variance of biological aging by genetics, for example, or gene-environment interaction, or air pollution, for example, or environment in general.
What about having a chronic disease? So having, for example, diabetes, does that, in your opinion, determine your biological age?
So we saw, on the contrary, we saw that biological aging, positive value of biological aging are associated with higher risk of first hospitalization and all-cause mortality. Determinants like diabetes and cardiovascular disease, for example, leads an acceleration in aging process.
Okay, so if I understand correct, you say if you have a chronic disease like diabetes, your biological age is lower than your chronological age.
Yeah, biological age is higher.
Oh, sorry. Yeah, so now I'm confused. Yeah, so that's higher.
Of course.
For sure. Yeah, so and that's also, I also do clinical practice, so when I see patients, and I see a lot of patients with diabetes, well, they look biologically older than their chronological age, and actually we think that having diabetes accelerates aging. And of course,
but you have to consider maybe the lifestyle of these patients can influence the biological aging. So maybe if they are smokers or if they...
Yes. Yeah, but on the other hand, for example, having chronic kidney disease, probably that, what do you think?
I think so. It's possible that this chronic disease, or maybe the drug, for example, can or the medicine, can influence. So, for example,
hopefully we decrease the age and not increase the age. And then, and you already explained in your introduction that you used a blood-based approach to determine biological age, and you said something about these neural networks. To be honest, I don't understand anything about that, but I'm not going to ask any questions about that. But what you said in your thesis, and also on page 88 of your discussion, you said, well, this type of network showed the best performance in predicting biological age. And well, while reading this sentence, I thought best performance in comparison to what?
In comparison to the other algorithm of machine learning.
I expected that.
Other algorithm and a classical statistic approach.
Yes. I'm just wondering, and then I come back to my first question. What was your gold standard? What is the best way of measuring biological age? Because you use this blood-based approach, and you say, well, you put it in a computer, and there is this network that works like human brains, and then there pops up an age. But how do you know that's the best biomarker of biological age? With what do you compare that?
Okay, there are other biomedical parameters used to compute biological age and of course consequently biological aging. So for example, if we use a brain age, for example, it's more accurately than the blood-based approach. But you have to consider that first of all, the blood-based approach can predict the chronological age of a subject. Other clocks can predict, so for example, mortality, but mortality is only one aspect of the aging. With this and
perhaps I have to put it in another way. So you have all these biomarkers. What did you predict in this? Did you predict age? Is that what you
chronological age?
Chronological
We
age.
predict the chronological age.
So that's what you did.
And our results was...
And if for one individual you did the prediction and that was higher or younger, then you said, okay, now I understand.
Biological age was accelerated or decelerated.
Okay, great. So now I understand. Okay. And then, well, then you did several analyses with different types of diets. Then I would like to go to... Well, wait. So if I look at the biomarkers, then I guess that these chronic diseases are already present in the biomarkers. Am I correct?
Mm-hmm.
Yeah. Because glucose was there, so I guess that with that you capture diabetes.
Yeah.
Yeah. And I saw that NT-proBNP was present, so with that you capture heart failure, I guess.
Exactly.
So I guess with these biomarkers, you more or less included different chronic diseases. And with that respect, I'm not surprised that by calculating this, you get a different type of age than the chronological age. Yeah. And then I wondered if I read, for example, just an example, chapter 6, let's go to page 82. Yeah, page 82. So, you did the analysis, and of course, what you need to do, you have to correct for confounders. And there, I read that you corrected for chronic diseases, CVD, cardiovascular disease, diabetes. So now I'm a little bit puzzled, because if biological age includes chronic diseases, why do you correct for that?
So, first of all, when we input the biological biomarkers, we use a standardization for these biomarkers. Moreover, when we added in the analysis this chronic disease, is because these can influence not only the outcomes, in our case biological aging, but also the exposure. Of course, the diet can be influenced by the diabetes or the hypertension or hyperlipidemia.
So that would be, of course, that's a definition of a confounder, that it
exactly.
Affects both exposure and outcome. But my question actually is diabetes not a part of the outcome? Is it not included in the outcome? And in that respect, is it not overcorrection what you did?
Yeah, but with the standardization, we delete this difference between each person, each individuals, using in the computation of biological age and consequently biological aging, of course.
I'm just looking at the program. Do I have time? Okay. So, well, this is completely clear. Thanks for that, because I'm a little bit less puzzled right now because of your answer, so I'm happy with that. Another difficult term you used is ultra-processed foods. Mm-hmm. Something very difficult. What in your opinion, and I read about your thesis, but what do you think? Okay, I think everybody is more or less convinced that ultra-processed foods are not healthy for you. What is it in ultra-processed food that makes it unhealthy to you? What do you think is the most important aspect?
Okay, so as I said, when we speak about diet, when we speak about food in general, of course, we have to consider diet quality. I mean, high content of sugar, high content of salt, of course, is not a good aspect of the food. But there are another aspect to take in account when we examine the impact of the diet on health. And this aspect could be the processing.
Yes.
So, the process, the ultra-processed food that we find in the last group of the NOVA classification are food products with intense chemical or physical processing. For processing, we means the altered food matrix, the content of chemical additives.
What happens then? So what makes it in the processing that it makes it?
Yeah, maybe they can alter the inflammatory biomarkers. So, for example, the neoformed compounds during the production of this food, such as the acrylamide, which is one of the most relevant components produced when the food are eaten to high temperature. The acrylamide, for example, was associated with high neurotoxic effect or increase the inflammatory biomarkers. On this can are associated with an unhealthy gut microbiota, or can may alter the glycemic response, and so on. So, for example, the presence of
what do you think about energy density?
Energy
density.
Okay.
That's actually, when I look at your table 1, and I look at the 5 quintiles.
Yeah, page, sorry.
83. And I see that energy density, so the energy ratio, I guess, that's the energy density, I guess, increases a lot from the lower quintile to the highest quintile. So that means, I guess, with every bite you take, you get much more energy
With the ultra-processed food.
So,
they are, yeah.
When you're full while having processed foods, you have had much more calories if you would compare that to the lower quintiles.
Yeah, and compared to the minimally, for example, minimally processed or unprocessed food,
Yes.
Of course, they contain much more calories, for example.
Per gram or per bite.
Exactly, for
Yes.
portion, yeah.
Okay, thank you very much. I'm very happy with your questions. I give the word back to you.
Thank you.
The opposition will be continued by Professor Shabanovska, Professor of Health, Leadership and Workforce Development at this university. Professor Shabanovska was member of the assessment committee, and she is the secretary of the degree committee.
Thank you, Prorector. First, I would like to compliment you on the thesis and, of course, your supervisory team. I've read your thesis with great interest. And my first question that I would like to ask is in relation to Mediterranean diet. And we can see through your thesis, and that's what's known, that there are clear benefits of having the Mediterranean diet. Since your study was carried out in Italy, I wonder, what can you tell us about the transferability of the diet to other regions, other countries with different climates and different eating patterns? And are you aware of any studies that dealt with this? What would you recommend? And the cultural adaptation, that it becomes sustainable?
Okay, so I'll esteem the opponent. Thank you for your compliments and for the very important question. So, as we know, Mediterranean diet, of course, it can be considered not only a shop list, but it can considered a lifestyle. Your question, your point is great, is very important. So, in the one of chapter of this thesis, not only we associated the Mediterranean diet with the biological aging, but other healthful, a priori healthful dietary patterns. So, for example, the DASH diet, and the DASH diet too is associated with the lower value of blood-based biological age. So other cohorts, not only the Mediterranean cohort. So, for example, in Spain, we found more or less the same results with other aging clocks, but also in other study, American or Occidental cohort, we find that the Mediterranean diet was associated with the lower value of biological aging. They measured the biological age in other way with the other aging clock, but the results was the same. In particular, with this thesis, and maybe in general with my research, with my work, I would that people do informed choices when they are at supermarket or restaurant or whatever. So the important things is to be able to choose what is the good products. So for a healthy diet. Of course, we know that the Mediterranean diet emphasizes the high consumption of fruit, vegetables, and cereals. With globalized food, with globalized in general, with globalized important dietary habits, maybe I think that so for or less, for all over the world, we can find the correct products to have a healthy diet, not only for the biological aging, but also for the cardiovascular disease.
So you are trying to say that it depends on the choice.
I think mostly. I think so.
On the choices. But are there any recommendations that, for instance, people in Finland or people in Scandinavian countries can use this diet for a longer period of time? Do you think that there can be some recommendations that they can stick to this diet longer for the prevention of?
Maybe in these countries they can choose fish, whole grains, and not only refined grains. So, for example, they can choose vegetables or fruit that, of course, they can find. They can choose. So, for example, speaking on the ultra-processed food, they can choose the bread and not, for example, the ultra-processed bread. They can find or choose fresh soup, vegetables, and not processed vegetables, so for example, processed food ready to eat. So just to know.
Thank you very much. And I would like to move to another question. I'm very happy that when I read your thesis, you showed a lot of associations and comments concerning public health. And in your proposition number 8, and may I ask your panelists to read the proposition?
So shall I stand up while I read? Or? Yeah. So the findings of this thesis may inform public health strategies at the population level by targeting groups of individuals more vulnerable to non-communicable diseases.
I wonder how.
So, this thesis could have a significant implication in biogerontology and nutritional epidemiology. In particular, with this thesis, we can be able to target the high-risk group at the population level of individuals, such as the elderly, who are more prone to develop chronic disease. This thesis showed the association between diet and biological aging, but specifically we examined the diet into two dimensions, diet quality and the food processing. The concept of diet quality, of food processing, and biological aging together could be considered a novel basis for a full recommendation of dietary strategies used to improve the quality of aging itself. Moreover, this thesis provided the evidence on the linking between healthful dietary patterns such as Mediterranean diet with biological aging, such as the active compound, for example, the polyphenols. Moreover, with this thesis, we show that the high adherence to a Mediterranean diet can lead a decreased consumption of ultra-processed food, and maybe this information could be used to settle new dietary guidelines to improve the beneficial effect of this healthful dietary pattern. Last but not least, of course, this measure of biological age, it's relatively easy to obtain, and in particular, these biomarkers are used in the clinical practice, in simple clinical practice, in the routine of clinical practice, and maybe this biological age, this tool can be used to screen the health status or the risk of clinical events, why not, in the general population.
Thank you. Can I have one more question? All right, so I would like to move on with the final question that I have for you. The benefits of diets like this are clear. And I would like to ask how public health policymakers can best introduce the benefits in education or in some recommendations that really the population is better informed and public health workforce knows how to do it. What would you advise?
So, in my opinion, at the school level, maybe can be used as a full recommendation. A new dietary guidelines can be explained in a better way at the school level. So, just to give you an idea to the children or adolescents to what is the best way to choose the product, to have a healthy lifestyle, of course, physical activity, diet, smoking status, alcohol consumption, and so on. So I think that the researchers, so the politician, have to speak with the people, have to explain which is our results, to give you the possibility to people to do an informed choices in health, so in field of diet, in field of lifestyle in general, and smoking status, and so on.
Thank you very much. I give the voice to the prorector.
Thank you. The opposition will be continued by Professor Stranges, Professor of Epidemiology and Biostatistics at the Schulich School of Medicine at Western University in Canada. Professor Stranges was also a member of the assessment committee. Professor Stranges, the floor is yours.
Yeah, thank you very much. And thank you, Simona, for the opportunity to assess your work. First of all, I would like to join the congratulations of the other colleagues for the quality, the amount of the work you have done over the last few years. So very, very well done. I have a couple of general questions, points for discussion with you, Simona. You already mentioned about obviously the influence of social determinants of health in terms of shaping dietary habits in the population. Also, obviously, the influence on your outcome, biological aging, as well as on the range of chronic diseases that obviously are affected by biological aging. You adjusted in your multiple studies. You are adjusted for the role of social determinants of health within the Moli-san study and also in the other survey, Italian survey based on phone interviews. But if you could you summarize, across the different studies, what was really the overall influence of social determinants of health on the observed associations? Did you find that in general people from the low socioeconomic status were more likely to, for example, have unhealthy dietary habits, and therefore they may be at increased risk of accelerated biological aging? Is that the consistent result that you found, or you did not really find a strong influence from the social determinants of health in your studies?
Okay. Highly estimated opponent.
My question is clear.
Okay. Yeah. No. You can hear me?
Yes.
Okay. Sorry. Okay. Highly estimated opponent. Thank you for your question and for appreciating my work. So, as you said, we adjusted in the analysis for the socioeconomic status, socioeconomic characteristics of the population studies. But in our cohort, in another study that I did not present in this thesis because it was a thesis based mostly on the diet, we find that the socioeconomic status in longitudinal analysis, we find that the socioeconomic status can influence the biological aging of the subject. In particular, we find that the changes, so to high to lower socioeconomic status, can influence the biological aging. This means that leads an acceleration in aging process. On the contrary, have a high socioeconomic status during the life can lead a deceleration in aging process, in biological aging, leads a negative value of biological aging.
Okay. So overall, basically, you corroborate the notion that there is also a social gradient in the association between, in general, dietary patterns and chronic disease outcomes. The second question I have is about talking about the Moli-san study. And obviously, you look at different dietary aspects of the Moli-san study. And I was wondering, what is the degree of variation and heterogeneity in terms of dietary habits within that sample from Molise region? So in other words, I'm just wondering about whether there was enough variation in dietary habits to really capture the potential impact of different dietary patterns or different, nutrients that you have looked at in relation to your outcomes. Or is this a more homogeneous population from that particular region with no really striking variations in dietary habits? So can you comment just on the degree of variation in diet within the Moli-san sample?
Yeah, so...
my question clear?
Yeah, I hope so. Okay, so we have to imagine that Moli-san study, of course, involved around 24,000 subjects from 35 years to 99, more or less. These can be, so our results correspond to the results of Italian observatory, cardiovascular epidemiological observatory that involves all the Italian sample. So another point is that the INES study, which we presented in this thesis, the degree of, so for example, of the Mediterranean diet adherence is similar, is comparable with the Moli-san study, and the INES survey involved all over the Italy and involved people from 5 years to 97. Around, we are speaking about around 9,000 subjects, around 9,000 subjects. Yeah. For this reason, I think that these are the study, the Moli-san study, the results of the Moli-san study could be comparable with the results, so for example, of the all over the Italy and why not of the other Mediterranean countries and populations.
Okay, I don't know if I have a time for an additional question, or I can stop here, whatever.
No, you have the time. You can continue.
Yeah. Okay, so Simona, so the last question I have is about the INES study, the telephone survey that is part of your chapter 5, and And so, because we know that in Italy there are important geographic variations in terms of life expectancy, in terms of occurrence of chronic disease. There is a north to south gradient. As we are aware, it used to be the other way around, but and again, we go back to the influence of social determinants of health. So you mentioned about that this was a representative sample of the Italian population, but obviously was a phone interview, right? And you mentioned that the socio-demographic profile of the INES study was comparable to that from, I think, the National Observatory, which I think is a fair comment. But did you look also at geographic variations, for example, in the consumption of ultra-processed foods across different regions of Italy within the INES study? I don't think that was part of the analysis, but maybe I have overlooked that aspect. So I was wondering whether there was any, consistent geographic variations in the consumption of ultra-processed foods across Italy.
Of course, we saw this different across not different region, but we divided Italy in center, north, of southern of Italy. In particular, we find that in the north of Italy, the ultra-processed food are most consumed compared to the southern of the Italy. But it's not a surprise. It was not a surprise for us because, as we said, the Mediterranean diet, the high adherence to Mediterranean diet, lead a lower consumption of ultra-processed food. And in southern of Italy, we expected that the strong food heritage in field of the Mediterranean diet can lead this high adherence to Mediterranean diet and consequently the low consumption of ultra-processed, of the dietary share of ultra-processed food.
So you're saying that basically your sample still confirms that stronger adherence in the southern part of Italy, because as you may be aware, there are also evidence from recent studies showing that in many southern Italian regions we have lost that adherence to the Mediterranean diet, and that may be one of the explanations why we have, adverse outcomes, even in terms of obesity or diabetes prevalence, seems to be higher now in the southern part of Italy than in the central northern part, and some of this is driven again by socioeconomic conditions. But it's just an observation, so I'm happy to stop here.
It's a good observation. So in particular in children obesity, unfortunately in southern of Italy we have a price. We had the price, so I know. But one of the limits, of course, of the INES studies, we are speaking about a study of 10 years ago. But the high adherence in the difference between ultra-processed between Mediterranean diets in the northern of southern of Italy. So we found this difference across the region, across the Italy.
Thank you.
Okay, thank you.
We continue with Dr. van der Meijden, Associate Professor at the Department of Biochemistry at this university. Dr. van der Meijden was also a member of the assessment committee.
Thank you, Mr. Prorector. Dear candidates, also my congratulations on this beautiful piece of work. And I especially like the cover because the woman is the symbol of youth, so that is always good, right? So I would like to go into the selection of the 36 biomarkers in the blood that you used. Of course, you can measure many different biomarkers in the blood. So can you comment on the way you have selected these specific biomarkers in the blood that you have used in your studies?
Sorry. So Thank you, esteemed opponents. Thank you for your congratulations, and I'm very happy that you like the cover of my thesis. So we started from the whole sample of the Moli-san study. We started from 41 blood biomarkers measured in our population. We chose these 36 for two reasons. So first one is we have to delete the 5 biomarkers that are, so for example, the total cholesterol, the complex, of course, the total, because we have to avoid the collinearity in the algorithm, in the computation of the blood-based biological age. Moreover, we choose these 36 blood biomarkers because these reflect the main biological pathway involved in the chronic disease onset and progression. With these 36 blood biomarkers, we can show all the domains of a body. So for this reason, as I said, and the other opponent, this could be this index of biological aging could be used as a tool to screen the health status, because it can describe all the domains of an organism, of a body.
Okay, so you mentioned that you have included the main domains, but it is known that the Mediterranean diet has a positive effect on health because of its antioxidant, but also its anti-inflammatory properties. But I noticed that in your selection of biomarkers, you only included one inflammatory marker, which is CRP, and no marker for oxidative stress. So that made me doubt whether your biomarker selection is really complete and whether you have indeed included the main pathways.
So we think, so compared to the other study, we think that this could be considered a complete way to compute biological age. So we have to remember that other cohort, other study, so other way to compute biological age, use less than 20 or less than 10 blood biomarkers to compute biological age. As we find in our study, the Mediterranean diet and polyphenols are associated in a positive sense with blood-based measure of biological age, but beyond their antioxidant effect. Maybe the association, maybe the mechanism that underline this association could be explained in other ways. So, for example, a healthy macrobiota or Ross scavenging effect. Maybe there are these kind of domains or these kind of aspects that the Mediterranean diet influence to decelerate the aging, delayed aging process.
Yeah. So in hindsight, would you now include other biomarkers to get a more comprehensive view on the more on biological aging? So would you include some additional biomarkers if you could do it again?
Okay, we tried with 15 blood biomarkers to compute biological age, and we associated this new screen, so say new screen, this new tool with ultra-processed food consumption, and the results was more or less the similar of the association found with ultra-processed food consumption and blood-based measure of biological age with 36 blood biomarkers.
So were these 15 new biomarkers? Or
No. Extrapolate among these 36.
So there are no additional biomarkers that you would add to improve the prediction? Or
No, not other biomarkers, blood biomarkers, but we are trying to compute biological age in the specifically organs. So, for example, the lung function or cardiovascular. We try to compute these kind of specific organ aging clocks.
Okay, thank you. Your subjects were recruited between 2005 and 2010, so that's already 15 to 20 years ago. Are the findings described in your thesis still relevant? Or should your conclusions be interpreted with caution, since the contribution of risk factors, for example, lifestyle, environmental factors, could have been changed over the years and over time? What do you think?
I think that the association that we found, so between Mediterranean diet and biological age, are validate, so you know, the association that we find. Of course, maybe there are changes to the dietary habits, but other cohort, of course, use the data of the 10 or 15 years ago, but the association is independent. The association that we find are independent by the passing of the time.
Okay. Yes. I have a question about chapter 2. And I find it interesting that the Mediterranean diet and the DASH diets, that they contribute to delayed biological aging, and there's a key role for polyphenol content. And you suggest that mainly the anti-inflammatory properties of the polyphenols impact the aging process. Can you speculate on the proposed molecular mechanisms behind these?
Yes, as we said, the polyphenols are involved in the mechanism that link Mediterranean diet, DASH diet, and biological aging, but beyond their antioxidant effect, because we saw that the total antioxidant content of these food dietary full are not associated with in this association. Maybe the Ross scavenging, for example, effect of the polyphenols, or the positive effect on gut microbiota, or the anti-inflammatory properties of this bioactive compound are involved in the association between this healthful dietary pattern and blood-based measure of biological aging.
Mm-hmm. And what would be the role of inflammation in biological aging in terms of mechanisms? Do you have any idea?
Yeah, so the inflammation are involved in biological aging. Indeed, a study in Italy, not of our group, found that we call it an inflammaging, which is the low-grade inflammation that occurs in the aging without apparently motivation, are involved in acceleration of biological aging, in particular in the cognitive decline of the subjects that are involved in that study.
Mm-hmm. And do you think there can...
Last question, or
yes.
maybe we can continue now. Okay, thank you.
Yeah. Last
So-
question? Okay.
Yeah, we can continue in the second round. Okay.
Yes.
So the opposition will be continued by Professor Spronk, Professor of Biochemistry of Atherosclerosis at this university.
Thank you, Mr. Prorector. Candidate, congratulations with this lovely thesis. I'm not going to say anything about the cover and the male and the female. Also, my congratulations to the team helping you in this. As always, I start reading a thesis from the beginning. Start with the introduction. And then already on page 8, I was reading that on Monday evening, and I was reading what the traditional Mediterranean diet is, and I read about unprocessed grains like pasta and bread. And I'm not going to challenge you whether that are unprocessed grains, pasta and bread, or whether they are processed. And then I got really feeling on my stomach that I got a taste of food, and I went to the local Quattro Mori, it's an Italian restaurant, one of my favorites, and I had my traditional prosciutto e melone, and I had my scallopino alla gorgonzola, which I flushed away with a little bit of Pinot Grigio. Only one bottle. One bottle. And my question relates basically to what was already hinted on by Professor Shabanowska. Am I now, according to my passport, 54 years old, or after this dinner, am I younger than 54 years?
So if you use it, highly esteemed opponents, thank you for your question. If you eat homemade scallopina, you are okay. It depends on the prosciutto that you use. So, for example, because there are some difference in if we are speaking about ultra-processed meat or processed meat. So, but when we use it, melone, you are in a safe.
So I'm now 50 years old.
Yeah, exactly.
Thank you very much. That was the answer I needed. But my question is more serious a little bit. You used the term Mediterranean food, and I'm still puzzling what is Mediterranean food. You mentioned unprocessed grains like pasta and bread, but yesterday I was also reading in our local newspaper an article on why the Italians are not so obese as the Dutch people. We eat a lot of potatoes, you eat a lot of pasta. And one reasoning was that the pasta, and mainly the dried pasta, contains the slow carbohydrates due to the processing of the pasta and drying. So, in your study, do you take into account all these variabilities in what pasta could be? Because I always learned from my Italian colleagues in Bergamo that you need to have grandmother who's making the fresh pasta, and that's the most beautiful pasta. We use the pasta from our local supermarket, Di Cerco, and think that that's the good pasta. But should I eat now dried pasta or fresh pasta of your grandmother? What is the good one, and do you take that all into account in your study?
Okay, when we speak about pasta, of course, so we have to consider that, of course, diet quality, of course, food processing, but we have to take in account the quantity, of course, of the pasta. Eat pasta every day is a good option, but of course, 150 grams of pasta is not maybe for everyone.
Okay, well, I'm disappointed in you. Exactly. Thank you very much. More in depth, on the same page, on page 8, that's basically where I stopped reading because I had a lot of questions there. You were talking about the telomere length. Mm-hmm. The telomere length is one of the hallmarks of aging, shortening of the telomeres, gives you a shorter lifetime. And then you mentioned a couple of sentences on, after that plant-derived foods can help maintain the telomere lengths. And then I start wondering, when I did my training, it was the area where they start cloning and have these stem cell technology available. And sheep Dolly was raised. And sheep Dolly died at the age of 2 years, predominantly due to the shorter telomere length. But as far as my knowledge on sheep goes, I always thought they eat plants predominantly. So how can you relate plant-rich food to maintaining telomere lengths and not shortening of telomeres like happened with sheep Dolly?
Yeah, of course, when we speak about biological age and biological aging, we have to consider, as I said, that this is the variance of biological aging is influenced by 48% by lifestyle. Of course, in this 48%, we are speaking about not only the diet, not only the plant food based or pasta or whatever, or soft drink in the other sense. We have to consider that we are speaking about all the lifestyle, smoking status, physical activity, for example, moderate alcohol consumption, and so on. For this reason, so we find that, or the colleagues found that plant-based food consumption are associated with good length of telomere length, but of course maybe it's not enough.
Okay, thank you. Then I have one maybe challenging question. While reading the introduction, I bumped into page 12, And you described the computation of the biological age, and Professor Brouwers hinted already on that, and that part was already clear to me. But then you state that the leukocyte counts with fractions summed below 99% or above 100% were set, 101% were set to missing. Can you explain that to me? Because you measure the total leukocyte count, and on the previous page you mentioned that that has a variation of roughly 4.2%, averaging on the cell count, which means for me that on average each white blood cell count will vary between 96% and 104%. So with that you would exclude a lot of cell counts and a lot of subjects in your cohort. Or do I misunderstand what you meant with this sentence?
No, we, as we write, we set the missing value. We set the missing value with a different way. So, or with an algorithm or with
Hora est.
Finish your answer.
or with the minimum or maximum imputation. Of course, it depends on they are below or above the limit of detection in the measuration of blood biomarkers.
Okay, thank you very much. I'd
You
like to
too.
give the word back to the program. Thank you.
Simona Esposito, the time appointed for defending your thesis has passed. The degree committee will now discuss the quality of your thesis and your defense. And I request that you and your company leave now this room. After the deliberation, we will come and collect you to return to this room.
Please all rise. Simona, we have in front of you and the two paradigms. Please follow me. The audience also, please follow me. departments. Please be seated.
Simona Esposito, the degree committee here present has assessed the quality of your thesis and your defense. In view of its positive verdict and taken into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor De Kate is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
Please all rise.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
I promise.
Thank you. By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Simona Esposito, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university.
Okay.
Congratulations.
Thank you so much.
There it is.
Yes, please be seated.
So Simona, congratulations. Let me hand this over to you. Secondant and then take the floor. And I'm happy to hand over the word to you. Second.
Just a few words for Simona. Simona Esposito started working with us in January 2019, joined the Platone project and other initiatives like Medicea in Preface, all epidemiological study at our department. Since in our department there was already another Simona, Costanzo, also a PhD from Maastricht University, we started calling her Simoncella, a cute nickname that in Dutch would probably sound like Klein Simona, or even better, Simonatie. Well, I will leave that to your preference, and sorry for my poor Dutch. Her PhD, which officially ended today, began 4 years ago and focused on nutritional epidemiology, with a particular interest in the relationship between diet and biological aging, a perfect topic for women that want to look younger than their ID says. Simoncella drives a small car, but in her mind, she's behind the wheel of a Ferrari. And in this illusion, she speaks at a supersonic speed, something you surely noticed during her thesis presentation. Maybe this speed has something to do with the fact that she comes from Pozzuoli, an historic town near Naples, known for its bradisism, a geological phenomenon that has been quite intense in recent months. So, like all Pozzuoli residents, Simoncella is always ready to very rapidly sound the alarm and make a quick escape if needed. She has also played a key role in recruiting participants from the Moli-san project and the Prevital project, other epidemiological cohort studies. She has already won multiple awards for Best Poster and Best Oral Presentation at various conferences. And just recently, she secured a temporary position at the Libera Università del Mediterraneo in Casamassima in Bari under the supervision of Professor Gianluisi, that is the expert in biological aging in our group. On behalf of myself and all the researchers at Neuromed, we wish Simoncella a long and successful career in the pursuit of elixir of youth. Thank you.
Dear Dr. Esposito, Also on behalf of the Maastricht University and the Board of Deans, it's a privilege for me to congratulate you with this new degree of doctor. The university highly appreciates this international collaboration, and in particular with the Moli-san group, and we hope that this can further continue in the future. Saying that, I believe that we also wish you a very successful scientific career, and I believe you have this afternoon demonstrated that you have all the skills to become very successful in your future career. Hereby I will close this academic ceremony. Before leaving the room, we will take a picture with all the members of the degree committee, including Professor Frenkels in front of the screen. And after that, we will move to the central hall and we will make a picture with all the members on site on the stairs in the central hall. Hereby I close this academic ceremony.
So please all rise, and may I invite everybody